Okay, so today we're going to talk about the malate-aspartate shuttle, glycerol 3-phosphate shuttle, pentose phosphate pathway, and the fructose metabolism. In order to explain the malate-aspartate shuttle and the glycerol 3-phosphate shuttle, we have to talk about ATP production. So when we go through glycolysis and the pyruvate oxidation and acid, when we go through all of these steps that will give us energy, we will either get 30 or 32 ATP. And if we get 30 or 32 ATP, depends on which of these shuttles, on which of these mechanisms that will transport NADHs through the intermembrane through the inner membrane of the mitochondria. So in glycolysis, we will produce 2 NADH, and these 2 NADHs, depending on how they get into the electron transport chain, depends on which transporter we will use. And that will give us either 3 or 5 ATP. Yes. So the malate-aspartate shuttle basically will... Okay, so we have glycolysis here. And glycolysis will give us NADH. And we want this NADH to be transported into the... we want it to be transported to the matrix of the mitochondria so that it can later be used in electron transport chain. But this molecule, we cannot just transport NADH from here to here. So what we do is that we convert oxaloacetate to malate. We use that by using malate dehydrogenase enzyme. And when we do that, we will kind of store hydrogen or electrons in the malate molecule. And the malate molecule, we can transport it through this malate alpha-ketoglutarate transporter. And then malate can be converted into oxaloacetate through malate dehydrogenase, and then we will produce an NADH plus H plus molecule into the matrix. So what happens here is not that the malate alpha-ketoglutarate transporter will not transport NADH from here to here, but it will it's kind of like a transport of electrons by malate, kind of like taking up the electron and then going in here and then doing the reaction, oxidizing. So malate will oxidize into oxaloacetate, and by doing that, it will give away a hydrogen to the NAD+, and then it will form NADH. Yes, and there will be the net ATP of this is 0, so no ATP will be produced or used. It's just the transfer of electrons that the malate-aspartate shuttle will do. Okay, so when we get oxaloacetate, the second problem is oxaloacetate cannot just diffuse through the membrane. Therefore, we need to convert oxaloacetate into aspartate. And we do that by the help of aspartate aminotransferase. So what happens is this aspartate aminotransferase will add a amino group to the oxaloacetate, and by doing that, it will take the amino group from the glutamate and it will form aspartate. So also this is important to remember that oxaloacetate, because I think for the colloquium we have to know how the molecules look like. So if we look at oxaloacetate right here, it has 1, 2, 3, 4 carbons, and it will take, because of the aspartate aminotransferase enzyme, it will take a amino group right here, and it will be put here, and we will get aspartate, which also has 1, 2, 3, 4 carbons and 1 amino group. So oxaloacetate and aspartate, the only difference is the amino group. And then when the amino group from the glutamate is taken, we get alpha ketoglutarate. So alpha ketoglutarate will look exactly like a glutamate, only thing is that it's minus the amino group, and therefore it will look like this. Hopefully that makes sense. Then when we get aspartate can be transported out of the matrix to the intermembrane space by the glutamate aspartate transporter. So basically aspartate goes out and glutamate enters. And it's really crucial that glutamate enters, because then that's when we can do the conversion of oxaloacetate to aspartate. Then once we get aspartate, we can once again do the same reaction, and then we will get back to oxaloacetate. And what's important to know is that these reactions on either side, from this side and this side, are exactly identical. Every single thing that's here will be here. So it's identical. Yeah, and this is a summary of that. So now we have the glycerol 3-phosphate shuttle. So it's it has the same idea of the malate-aspartate shuttle. Only thing is that we will start with, so glycolysis will give us NADH plus H plus. And then what happens is that the dihydroxyacetone phosphate molecule will take up these hydrogens, will accept these hydrogens, and then we will get glycerol 3-phosphate, which is this molecule. Which is this molecule right here. And then glycerol 3-phosphate will will donate its hydrogens to an FAD, and the FADH will donate its hydrogens to the ubiquinone, so it will become ubiquinol, and then it will enter the complex 3 of the ETC. What's important to know here is that we have given away, or we have, like, from glycolysis we have an NADH that has been converted into FADH. And remember that NADH gives us 2.5 ATPs from electron transport chain, and FADH will give us 1.5 ATP. So when we convert NADH to FADH, that means that we have we gain 1 ATP less from this process. But this is beneficial maybe if we do fast movements, like in the skeletal muscles. And the electron transport chain here will be activated from the outside of the intermembrane space rather than from the inside, and this saves a lot of time, and this saves a lot of time compared to the malate-aspartate shuttle. So if we start from the outside, it will go faster, but we will lose 1 ATP. Yeah. Oh yeah, another thing I wanted to say is, here we have also glycerol. So after fatty acids break down, we will get a glycerol, and glycerol can actually enter this glycolysis through the glycerol 3-phosphate shuttle. Yes, and this shuttle, glycerol 3-phosphate shuttle, is really important for memory, and lack of it leads to intellectual disabilities. Okay, now we will go to the pentose phosphate pathway. So this doesn't have to do anything with the shuttles. Important to know for the colloquium is that we actually have to know all of these intermediates in this reaction or in this pathway. So we have to learn all of them. Okay. So the pentose phosphate pathway will lead to the production of NADPH and DNA intermediates. That's the main thing. And it will happen in all of the cells in the cytosol, because that makes sense. Like in every single cell you will have DNA, and you will need to build up this DNA by the pentose phosphate pathway. Secondly, you will need NADPH, and we will talk about why we need NADPH, but okay, so we will need NADPH in every single cell, and we will need DNA. And so this will happen, the PPP, pentose phosphate pathway will be happening mostly in rapidly dividing cells because they need a lot of DNA, they need NADPH for fatty acid synthesis, etc. And also cells with high amounts of ROS, so reactive oxygen species, will also need a lot of NADPH because NADPH can help neutralize these reactive oxygen species. And also this reaction, the pentose phosphate pathway, does not make or use ATP. That's important. And the glucose and the pathway will start from glucose 6-phosphate and end with ribose 5-phosphate, or earlier, we will talk about it. Yes. We don't have to know exactly the difference between NADPH and NADH, but I think they said it in a lecture earlier, in the lecture of citric acid cycle. It was mentioned that NADPH plus H plus exists in the cytosol, and NADH plus H in the mitochondria, but obviously, like, things can diffuse in and out, or not diffuse, but yeah, like they can exist in both, but we don't have to know the difference. So, purpose of the pentose phosphate pathway, we just talked about it, is the production of the DNA and NADPH plus H. Also, NADPH will not go through electron transport chain, therefore there will be no net gain or loss of energy. So the products mainly, as I said, is NADPH and ribose 5-phosphate. Ribose 5-phosphate is actually the precursor for nucleotides for DNA molecules. And we will use this NADPH molecule for the reductive biosynthesis. Okay, so reductive biosynthesis means use of NADPH, and we will use this in fatty acid synthesis and synthesis of cholesterol and steroid hormones, and also for repairing oxidative damage that is caused by the reactive oxygen species. Also DNA and RNA synthesis and synthesis of ATP, NAD, and FAD. And this pathway, as I said earlier, happens in rapidly dividing cells. So for example, bone marrow, skin, intestinal mucosa, and also tumors. We have to remember this also. When we convert, so we will start with glucose 6-phosphate, and then we will go to 6-phosphogluconate. And then when we go from 6-phosphogluconate to ribulose 5-phosphate, we will produce a carbon dioxide. The pentose phosphate pathway can actually happen either to here, like it will go from glucose 6-phosphate to ribulose 5-phosphate, and then it can actually go back to glucose 6-phosphate. And that's because maybe we only want NADPHs. We don't want ribose 5-phosphate, because ribose 5-phosphate, as I said earlier, is good for the production of DNA, RNA, and ATP, NAD, FAD, and the coenzymes. Yes. And the two phases of pentose phosphate pathway is the oxidative phase and the non-oxidative phase. And we will talk about, so oxidative phase will go from glucose 6-phosphate down to ribose 5-phosphate and create these DNAs. And non-oxidative phase will just go back to glucose 6-phosphate. So the purpose of the non-oxidative phase. So it depends on what the cell needs. If we need ATP, for example, then we will go back to glucose 6-phosphate, and then glucose 6-phosphate will then be converted to fructose 6-phosphate. So then it will enter glycolysis. maybe we want to store energy, so we will go back to gluconeogenesis. Or maybe we only want NADPHs, so therefore we will need to go back to glucose 6-phosphate and to generate NADPH. Okay, so that's the purpose of the non-oxidative phase. Okay, and the end here, we will get ribulose 5-phosphate. Ribulose 5-phosphate can be converted to ribose 5-phosphate, and ribulose 5-phosphate can either become ribose 5-phosphate or another molecule called cellulose 5-phosphate. So when we want to go back from ribulose 5-phosphate back to glucose 6-phosphate, what will happen is that we will have this cellulose 5-phosphate together with ribose 5-phosphate, and we will get glyceraldehyde 3-phosphate and ketoheptulose 7-phosphate. I will not go through every single reaction because they do that in the lecture, but what we have to know, and what's important for the clock according to the guidelines, of course, is that a transketolase is, for example, like this enzyme and this enzyme are important for transfer of 2 carbon units. So when we want to go back from ribulose 5-phosphate to glucose 6-phosphate, we will do these reactions, and we will need to, so when we transfer 2 carbons, we will use a transketolase, and when we use transketolase, we will use a TPP, so the B1 vitamin. And when we transfer 3 carbon units, then we will use a trans-aldolase. Here in this reaction, energy is not used. This is only a conversion between 5 carbon atoms until we get a 6 carbon atom, because at the end of this we will get the fructose 6-phosphate, and the fructose 6-phosphate can then go back to glycolysis, or it can go to gluconeogenesis, etc. So just remember the purpose. So the main thing from this is the purpose of the non-oxidative phase. It's this: we do not gain energy or lose energy, and please remember what the enzymes do. So regulation of the pentose phosphate pathway. The purpose of the pathway is to create NADPH plus H+. So if we have a lot of it, then the pathway will be inhibited, and it will be inhibited by the NADPH plus H+, and it will inhibit an enzyme called glucose 6-phosphate dehydrogenase. Glucose 6-phosphate dehydrogenase helps the conversion of glucose 6-phosphate to 6-phosphogluconolactone. So if we inhibit this enzyme by NADPH plus H+, then the pathway will be inhibited. In cancer, though, there will be an overexpression of this enzyme, and an overexpression of this enzyme will lead to a generation of a lot of NADPH plus H+, and also a high generation of the ribose 5-phosphate, so that the cancer cells can make DNA, and also fatty acids. And so patients with overexpression of this enzyme also have very poor survival rates. Okay, deficiency in the PPP enzymes can lead to oxidative stress. So the enzymes of the pathway, they produce NADPH plus H+. And NADPH plus H+, like I said before, they're important for fatty acid synthesis, or many stuff. But also they help in reducing reactive oxygen species. So let's take a look at this. You have oxygen. Oxygen might become a superoxide radical. This superoxide radical will be neutralized by addition of 2 hydrogens, and this will lead to the production of hydrogen peroxide. But hydrogen peroxide also needs to be neutralized, and we can neutralize hydrogen peroxide by glutathione peroxidase enzyme. But this glutathione peroxidase enzyme can only work with the cofactor GSH. So GSH will donate one of its hydrogens to the hydrogen peroxide and produce water. But when that happens, GSH will become GSSG. And so the role of NADPH plus H plus in this is that NADPH plus H plus will actually give away hydrogens or electrons to the glutathione reductase. Glutathione reductase will accept these hydrogens, and then it will donate these hydrogens to the GSSG. So GSSG will receive the hydrogens from the glutathione reductase, and glutathione reductase got these hydrogens from the NADPH plus H plus. And so when that happens, we will get GSH. GSH can then help glutathione peroxidase to neutralize hydrogen peroxide. And this is a summary of what I just said. So basics of fructose metabolism or fructolysis. So fructose metabolism, and also we will talk about this in lecture 6 about galactose. They will have to enter a glycolysis pathway in order to be metabolized. So basically they will have to be converted into an intermediate of glycolysis, and then they can enter the pathway of glycolysis, and then they can be metabolized. So fructolysis happens in the cytosols of human cells, but mainly it will occur in the liver, mainly in the liver, but also kidneys and the small intestine. If we eat normal amount of fructose, then it will be metabolized in the enterocytes, and it will enter the enterocytes through the GLUT5 transporter, and then it will be converted into glucose, and then glycolysis will happen. So basically this is what we see in the picture: fructose or high amounts of fructose, then through the GLUT5, then fructose will be converted into glucose, and then glycolysis. But if we eat very high amounts of fructose, then they will be metabolized in the liver. 50% of fructose will be metabolized by the liver. Or it will be metabolized by the muscle, but there are different enzymes that work in the liver than in the muscle. Yes, and we have to remember GLUT5 for the colloq and GLUT2. GLUT2 is for the transport of glucose, GLUT5 is for the transport of fructose. Okay, so Fruktokinase C and Aldolase B are essential for fructolysis in the liver. So when we have a fructose molecule, we have to convert it to either fructose 6-phosphate or fructose 1-phosphate, and this depends on which enzyme is available in a specific cell. So a hexokinase, for example, can convert fructose to fructose 6-phosphate, but the thing is that hexokinase has low affinity to fructose, so it's not that effective. And fructokinase A, which exists in the skeletal muscle, has low affinity to the fructose. It can convert it to fructose 1-phosphate, but it has low affinity. So there needs to be very high concentrations of fructose so that muscle can actually metabolize the fructose. Lastly, we have fructokinase C, and this we have to remember. Fructokinase C, which exists mainly in the liver, will be able to convert fructose to fructose 1-phosphate, or phosphorylate fructose to fructose 1-phosphate. And when that happens, the next step would be to cleave fructose 1-phosphate, and the cleavage of this molecule can be done by aldolase B in the liver. And this step is the slow step, so it's also the rate limiting step. And fructose, okay, we'll talk about this soon. But just remember, fructokinase C and aldolase B are important for the fructolysis in the liver. In the liver, fructokinase C and aldolase B. Okay, now we can go through this again. So we have fructose. Fructose together with fructokinase, so this is in the liver, with fructokinase C in this case, this reaction is very, very fast. So this reaction is very fast. And then we want to cleave fructose 1-phosphate. The next reaction will be slow. So this means that there will be an accumulation of fructose 1-phosphate. And this accumulation actually can lead to the activation of glucokinase. And glucokinase is hexokinase 4. So it's an enzyme in the liver that has low affinity to glucose. But fructose 1-phosphate can activate glucokinase. When glucokinase is activated, the glucose can actually be converted back to glycogen. So that can happen. But when we cleave, so let's move on. When we cleave fructose 1-phosphate, because fructose only has one phosphate right now, so when we cleave it, only one molecule will have a phosphate, and so we will get dihydroxyacetone phosphate, and we will get glyceraldehyde. And this glyceraldehyde, together with the trio kinase, we can actually make it into glyceraldehyde 3-phosphate. And when we have that, when we have glyceraldehyde 3-phosphate, that means that we can either go back to gluconeogenesis, or we can go to glycolysis pathway, depending on what kind of needs the cell has. Then we have fatty liver disease. So if we have consumed a lot of fructose, and our glycogen stores are full, like we have already stored fructose, the glycogen stores are full. What happens is that fructose will be converted into acetyl-CoA. And when we have acetyl-CoA will actually be enter the fatty acid synthesis, and this will cause fatty liver disease. So diseases that are caused by enzyme deficiency in pentose phosphate pathway. So we have lack of enzyme fructokinase, also called fructosuria. So fructose, in order to be converted to fructose 1-phosphate, we will need enzyme fructokinase. But if we lack this enzyme, this will just lead to fructose being excreted with urine. And this disease is benign. The second disease is lack of aldolase B. And a lack of aldolase B is called hereditary fructose intolerance. So if we don't have this enzyme, this would mean that we will have an accumulation of fructose 1-phosphate. And this is bad because the phosphate group here will not be, we need this phosphate group for other pathways. And if this phosphate is not let go from this molecule, then a lot of pathways will not be able to function. So for example, oxidative phosphorylation cannot happen if the fructose 1-phosphate is holding on to that phosphate. And also this phosphate is important for glycogen breakdown, which is the next lecture, and gluconeogenesis. So this is what can happen, and for people with this disease, they just have to avoid fructose. It's bad. I don't think there is a cure. Yes. So basically, it will stay attached to the It will stay attached to the phosphate. So fructose metabolism versus glucose metabolism. Fructose metabolism will occur primarily in the liver by enzymes fructokinase C and aldolase B, whilst glucose metabolism will be mainly broken down, or it will mainly occur in muscle cells, and the enzymes is mainly hexokinase and aldolase A. For ATP calculations, what you have to know is actually it's going to be the same. So in glycolysis, we gained 2 ATP plus 2 NADH, and it's going to be the same for fructolysis. It's the same ATP calculations. We will invest 2 ATPs. The thing is that the investment of 2 ATPs will actually occur here. So in the liver, we will, when we convert fructose to fructose 1-phosphate, we will invest 1 ATP, so that's -1. And here we will also invest -1 ATP, so that's -2. And then it will enter glycolysis from the GA3P. And then the calculations will all be the same. And if glycolysis has +7 ATP, then fructolysis will also have that. It's gonna be the same. Okay. Yes, that was it for the for this lecture. Thank you.